we've lived through 2 units. So we are starting on cells, and even though we have this packet, and we will answer this little question, this is actually something we need to do like a whole video on. So I'll just start off with no glue in, and we'll just start taking notes. And so this is the cells unit. What do you notice? Notice I'm in a whole new notebook. Wow, I'm ready for a whole new notebook. Crazy. So this is the cells unit, and this will feel more comfortable to you, I think, than what we did with the biochemistry pieces. And what we're going to spend the time in this video making sure we understand is the difference. Like, what is it to be a prokaryote compared to, so versus, not like they're fighting, eukaryotes. So there's a whole lot of things that you can spend some time doing as far as if you studied about this. Prokaryotic cells are old. Like, they're the original cells. So I just usually, because you know how I am, I don't like to write. So I'll just put things like that so we can just talk about those. So prokaryotic cells, they're the original cell. So as life developed the ability to replicate itself, feed itself, and that made us recognize that it was living, movement, all of those traits, the first cells were prokaryotic. The cells that are left that we have on the planet that are prokaryotic cells, so these are the original cells on Earth. Right now, if you think about it, it's going to include, so prokaryotic cells would include bacteria and archaea. Now, you don't have to know, we're going to look at domains later and we'll walk through these. Right now, we're just saying this life still exists, obviously, prokaryotic life, and we would see it as bacteria or archaea. So I like to think about it this way. When you're comparing the two, so let's kind of just make the obvious point here. Eukaryotic cells, they came later. So they came later, and I guess what we'll call Earth time. So they're after prokaryotic cells, and all of the things that would be on Earth that you would think of as eukaryotic cells would be you, right? So it would be animal cells, plant cells, and then fungi, so fungus falls into this too, protists, which you may or may not know what those are, algae, protozoan. So any of those would fall into eukaryotic cells. So an easy way to begin to think about them is to start with size. I think that gives perspective of what changes there are. So before we even start, think about the difference in the job of being the mayor of, let's pick something tiny, like Abingdon, little bitty town Abingdon, compared to the mayor of New York City. What's the difference? Well, managing something much larger is much more complicated. So size gives you an indication of complexity. So here's the way we'll work with it. Eukaryotes range in size from 10 to 100. These are called angstroms, but they're micrometers. And it's purposefully with that funny little piece there. Prokaryotes range from 1 to 5 micrometers. Much, much smaller. So if we can get a perspective of that, you know, let's just pick something. Let's just go with like a middle here. So if we let, I don't know how to do this, but I want to do it. I guess I could use my ruler. Okay, so I can fit this on there. So what I'll do is I'm going to make something that's 10 centimeters long. So 10 centimeters, each centimeter has 10 millimeters. So this is 100 millimeters. And this is just an example for perspective. We will let that represent, so we're going to put here that this represents 100 angstroms, 100 micrometers, which is the size of a eukaryotic cell. That would mean, I don't even know if I can draw this, that this right there, this is 1 millimeter, and we're going to let that represent 1 angstrom, 1 micrometer. So this right here is the prokaryotic size, and this whole piece right here is the eukaryotic size. So prokaryotes are super tiny compared to eukaryotes. Much, much smaller cells. So size-wise, prokaryotes are tiny. Compared to eukaryotes. 1/100th, 1%, tiny compared. So obviously we would make this more three-dimensional or whatever, but this gives us perspective of just, there's a huge difference in size. Size again, increase in size, most of the time biologically, at least when it comes to cells, is going to be the opportunity for an increase in complexity. So what does a eukaryotic cell look like? Now I'm going to draw one, and I think what I'm going to do is I'm going to decide that I'm going to let this whole page be a eukaryotic cell, just because I want to keep scale in our head. So I'm going to put eukaryotic cell here, and I'm going to go ahead and just give myself all of this as a boundary, literally the whole paper. Then I'm going to come over, and if you think about it, it would be maybe this big for a prokaryotic cell. I bet you could fit 100 of those in here easy. So that's about the difference between the two for size. You can actually put what we need to draw right in here. So prokaryotes, they don't have sections. So I saw a really, really good analogy, and I've usually done another one, but I don't think it's near as good as this one. So if you think about prokaryotes, they are like, well, probably what I could afford if I lived in New York, or maybe even California. Like the tiniest single room efficiency apartment. So they are like a tiny single room And if you don't know what an efficiency apartment is, I'm going to explain it. So that's what prokaryotes are like. Eukaryotes are like a McMansion. So it's not just size that's part of the difference, teeny tiny, freaking enormous, but it's also the number of rooms. A single room, okay, we got that. Efficiency apartment means in one room you're going to have a little kitchenette, a little bathroom area, a place to sleep, and a place to sit. That's it. Every single thing that would go in a regular apartment has to go in this one room, tiny little single room. So there's no compartments, there's no separate living areas, there's no separate nothing. Okay, versus in a McMansion, where you might have 2, 3 kitchens, 3 or 4 different living areas, 7, 8, 9, 10 bedrooms, 10, 11, 12 bathrooms, 12 garages. I mean, you seriously have space for everything. So this is the difference between the way that they're put together. But neither one's better than the other. It's just 2 different strategies on the way that things can be successful. You can either keep it super simple, or you can make it very compartmentalized. There's benefits to both. There's drawbacks to both. So this is the size of what we would get. There's not going to be any rooms, compartments in here. Whatever goes in here as a prokaryotic cell goes in here just like that. So let's think about what we've got to manage to fit in there. I'm going to make a list. So for the prokaryotic cell, we need to put what's called a nucleoid region. Definitely do not call that a nucleus. It's not. It's just like in the single room efficiency apartment. If you've got a chair and a little table, that does not make you have a living room. Okay? It's not going to be compartmentalized. But it's going to be the area where the DNA is. And so you're going to have circular DNA. It's not in the nucleus, it's just in the cell. Everything's in the cell, loose. So I'll draw that. I'll see how I can do. I might have to get my little fine pens to write what these things are. So that's the circular DNA. So maybe I'll just come over here and put, hey, that's what that is. Then they have one organelle. Where's my finger? Right there. One organelle. It's not membrane bound. Ribosomes are not membrane bound. They have ribosomes. That's it. That's their organelle they've got. And so we'll just draw those like this. So I'm going to put some ribosomes in there. Prokaryotes, bacteria, have a cell wall, so it's the rigid structure. It's made of a polysaccharide, but it's not like the cell wall of plants, because hello, it's not a plant. Plants are eukaryotic, these are prokaryotic. They have a cell wall, and it's called peptidoglycan. And it is a polysaccharide, just like cellulose is. It's just a different structure. So there's my cell wall. Not all Some of them have a capsule. We'll put some. This is also carbohydrate, but it's kind of gooey. So think of it kind of like snot. And so there's a capsule outside of everything. So there's my capsule. Some of them have it, some of them don't. And we'll go into what all this stuff is when we get to like their structure. Right now we're just comparing them. They also have, they call them two different things. They're called fimbria. That's the plural. They're just little hair-like structures. Definitely people call them cilia, just so you know. Then they have something called pili. This is a much less hair-like. This actually is almost like a straw. So we'll put that it's a straw-like outer feature. I don't know what to call it. But they'll send, I'll bet you didn't know bacteria had sex. They do. They'll send DNA through this straw-like feature and share it with each other. So we'll draw that. So we'll put one like right there. Then they can have flagella, which we know what those are. They're like whip-like. So they're long, whip-like attachments. So that would be the flagella. But that's it. What'd you think? You're getting a whole lot more for your efficiency apartment? Hello. Nope. Bare bones basics. That's what you get. That's very, very different. That's a different way to live. Super simple. Pretty sure if you lived like this in New York City, you could pack everything you owned and carry it with you and move someplace else because you're just living light. That's not going to be the way people live in a McMansion. That's not the way eukaryotic cells do business. So we'll come over here, and now let's do our eukaryotic cell. So, wait, did I put cell membrane? Let me put that on here. They do have a cell membrane that's going to be inside this cell wall. So I'll draw it like this. Let me make sure I've got this for sure, like, you know, for real. And so I'll draw my cell membrane in like that. That's pretty good looking. Totally different mindset with eukaryotic. Eukaryotic, everything has its own separate space. So, you know, if you have a McMansion, you might have a dog bedroom bigger than this whole apartment. You know, you could have all kinds of things. So you're going to have, eukaryotic has membrane bound, which we're going to work on what membrane means. So surrounded by membrane organelles. And they're called organelles because they're little things that have one job. Just like you have a heart and it has one job. And it's an organ. So organelles are the same thing. But these have a membrane. So eukaryotic cells have, so we come over here, put them in there. They have a nucleus that has a double membrane. So I have plenty of room, I'm going to draw them. So you've got a nucleus that has a double membrane. And inside the nucleus, you're going to have DNA, but it's not circular. It's linear. So here's your DNA. This is the nucleus. That's the double membrane. Also, so can I kind of follow along here, all living things have ribosomes. That's the one thing everything has in common. If you're a living thing, you have ribosomes. Ribosomes are where proteins are made. You can't be living without proteins. So they also have ribosomes. Their ribosomes are a little bit bigger, so I'll make them bigger. So you've got ribosomes that are attached to this endomembrane structure that comes off of the membrane, off the nucleus. So you've got, this is your endoplasmic reticulum, which is the ER. It's just a membrane extension of this. And the reason why you have so many membranes is a lot of different cellular processes have to occur on a membrane. So you put them in there all like this and increase the surface area, and you have more surface area for the processes that need to happen on the membrane. So there's the endoplasmic reticulum. Some of the ribosomes are attached that makes this the rough ER. And then some of the endoplasmic reticulum, so I would have, let's put these out here, more ribosomes. And then I have some ER that doesn't have it, so any ribosomes. So this is the rough ER. This is the smooth ER. It's an S. So we'll come over and I guess I'm needing to, I'll put this over here in just a second. Let's stay in the groove. Some eukaryotic cells have a cell wall. Whoops, there's supposed to be a wall. That would be plants. It's cellulose, which is also a polysaccharide. This is the cell wall, and then here is the cell membrane. So if you're a plant, you have both. If you're an animal, you only have the cell membrane. There's no capsule, no gooey weird thing on the outside. Hair-like structures, sometimes you have cilia, so there's the potential that you could have cilia. So there could be. Those would be like the cells that line the inside of your throat. And that's why that you're able to always sweep up the mucus from your lungs that goes up into your throat and swallow it down. So, cilia. No sex pill eyes. They don't do that. Some have flagella. Hello, sperm. That's one. So, this would be one, so flagellum, but they do have them. They've got those. They have a cell membrane. So up in here, where we've got space, I'll put they have an endoplasmic reticulum. All that is just a bunch of membranes inside. And then everything that's inside of here is going to be cytoplasm. Prokaryotes have cytoplasm. It's just everything inside the cell membrane. You've drawn these before. So we have a double membrane nucleus. You have mitochondria, where cellular respiration occurs. Notice how everything is its own little compartment, just like a room in our McMansion. I mean, I don't know about you, but like, I don't want to go take a shower in front of everything. Like, if you got company, where are you going to do it? You're going to do it in front of everybody? No. I'd kind of like to have little compartments, because a shower is a specific location to do a specific thing. And so that's what eukaryotes have. So I'll draw another one. Okay? Notice they're pretty much the size of a prokaryote. Then you could have a chloroplast if you were a plant. So mitochondria are in plant and animal cells, both. Maybe I should. Yeah, both. Don't be me. Animal and plant cells, both. All eukaryotic cells have mitochondria. Get over here. Mitochondria. Plant cells have chloroplasts. Both of these have a double membrane. So if you're a plant cell, you're going to have a chloroplast. So I'm just going to draw it with some flattened discs inside. And then I'll just put a couple more. So we have a couple of chloroplasts in there. Cytoplasm. Ooh, vesicles. So you've got little membrane bound, because that's what we're talking about. Everything's in its own package. So this would be a vesicle. You've seen some, so like we'll put example like a lysosome. That would have a very unique environment that's low pH, like to digest stuff. So this is going to give you the ability to have little microenvironments. So that would be vesicles. And how are you going to manage all this? Do you just throw up the outside and don't put any structure on the inside of your McMansion? I bet not. So you're going to have structural all over, like all over. So this would be the cytoskeleton, and you'd have all kinds of structural different pieces. So you got to hold your great big thing together and hold it apart and make it have some actual structure. So that's the big thing about what the difference is. So all of these are their own individual, if you think about it, rooms. You have a vesicle room. Well, do you do something different in the vesicle room than you do the nucleus room? Well, yeah, because I called it a vesicle room. If it was the same as the nucleus room, I'd have called it a nucleus room. They're different. Each one of these lets you have individual events happening and little different conditions happening because they're separated. So you have all these separate compartments. So that's the key to this. You have many separate compartments. Which equals the ability to separate different processes. So you can separate many processes. Because I don't know if you've been someplace like this before, like so fancy, you're uncomfortable. I have. I had friends whose families, I mean, I don't even know how they lived like they lived, but their houses were huge. I lived in Memphis, and I mean, they like had a compound. They had barns and horses, and they had lakes, and they had a house, and they had a carriage house, and they had guest house, and they had a place for people who worked for them's house. And their houses were as nice as my house. And when you go in here, it's so, I don't know how to describe it. Like, there would be dinner, but you didn't go to the kitchen, because that wasn't your job. That was for the people who cooked. So, I mean, there was obviously a place where the food was prepared, but it was so separate from me. Like, I didn't even know what happened there. It was that separated. That's what I'm talking about. So, what happens here in this mitochondria is so separated from the rest of what's going on. It's like its own little thing. So you have to also have the ability for cells to coordinate, because, I mean, somebody told these people to bring dinner to us, and so there was some type of coordination. So this is going to require communication. Well, I don't know. And it also requires coordination. Goodness, here you might be able to sit in your little chair where you're eating dinner and reach over and open the oven and get it out. And then when you're done, reach over and put it in the sink and never get up. Like, there is no separation. So this is the key to this one. It's pretty simple. You don't have separated processes. They're all happening in the same space. So, you know, you better not have things that are too complicated that have to have their own little, like, lysosomes, low pH. You don't want to digest yourself. Well, that's not happening over here, because you don't have any separate spaces. So there's no separate spaces. All right? So at some point we're going to talk about the endosymbiotic theory, and it's just the theory that these prokaryotic original first cells, once there were eukaryotic cells, the eukaryotic cells engulfed some prokaryotic cells that were able to do cellular respiration. So they're saying mitochondria, there's strong evidence that it was once free-living prokaryotic and not just an organelle. And chloroplasts. Once that there's evidence that chloroplasts used to be free-living prokaryotic cells. So don't worry about that too much. We'll talk about that. But it gives you perspective when you draw it like this, that I can see that. Like, you do something super cool. Yep, I do cellular respiration. Bingo. Engulf you, and now I can do cellular respiration too. So, anyway, I hope that makes sense. I'm going to put some other little people talking. I know the Amoeba Sisters are kind of silly, but their stuff's really good. So that's what's coming next, and I think you're wonderful.
I'll never forget a circular red spot I developed on my arm when I was in elementary school. It left a lasting memory in my mind because it was something called ringworm, and with my active imagination, I thought I was now infected with a ring-shaped worm. I learned you gotta be careful about names because ringworm isn't caused by a worm at all. It's actually a fungus, which it turns out is pretty common and can be carried by many things like pets or soil. And since up to that point I was used to antibiotics as a way to treat infections, I assumed I'd be given antibiotics. But I wasn't. I was given an antifungal cream instead, and it went away. So it made me wonder, what made it different from the bacteria that had made me sick in the past? Why wasn't I given antibiotics? Well, antibiotics target bacteria. Antibiotics can destroy bacteria by affecting their ability to reproduce, damaging their cell walls, or interfering with their ability to make proteins that they need to survive. Just some examples. But it turns out bacterial cells and fungal cells are very different cell types. In fact, fungal cells have more in common with your cells, which are animal cells, than they have in common with bacterial cells. And that has a lot to do with the comparison of prokaryotic cells with eukaryotic cells, which is what we will focus on. First, just a refresher, recall that the modern cell theory includes a statement that all living things are made of one or more cells. All living things. In the three domains of life, prokaryotes are organisms that can be bacteria or archaea. They are unicellular, which means they are single-celled organisms. Eukaryotes are organisms that fit all in this last domain, eukarya. Eukaryotes can be protists, plants, animals, or fungi. They can be unicellular or they can be multicellular, which means they can be made up of many cells, like you. By the way, just to clarify, the word prokaryote is typically used to refer to the organism itself. When you are describing its cell, you are describing a prokaryotic cell. Same for eukaryote. Eukaryote typically refers to the organism itself, and when you are describing its cells, those are eukaryotic cells. Prokaryotic cells and eukaryotic cells do have a lot in common. Both have DNA. That's critical because DNA is the cell's genetic material. Both prokaryotic cells and eukaryotic cells have ribosomes, which are small organelles, an organelle being like a tiny organ. The ribosomes have the important job of making proteins. Gotta make protein. Both cells have cytoplasm, the jelly fluid within cells. Both of them have a cell membrane, also known as a plasma membrane, which is critical because it controls what goes in and out of the cell, and therefore maintaining homeostasis. All cells have a cell membrane. Now, as for cell walls, most prokaryotic cells have cell walls. Many eukaryotic cells, plant cells and fungal cells, for example, can have cell walls. But there are plenty of eukaryotic cells that don't have cell walls, such as animal cells. What makes prokaryotic cells and eukaryotic cells different is especially interesting. Eukaryotic cells are more complex than prokaryotic cells. They tend to be larger than most prokaryotic cells. And to help me remember some more differences in this next part, I like to remember that pro in prokaryote rhymes with no, and eu in eukaryote rhymes with do. Prokaryotic cells have no nucleus to contain their DNA, so you will find their DNA is not contained within a nucleus. It's a bit messy here. They have no membrane-bound organelles. Membrane-bound organelles are fancy organelles that have their own membrane, like the nucleus, mitochondria, the endoplasmic reticulum, and the Golgi apparatus. A big indicator of eukaryotic cells is this nucleus. Eukaryotic cells do have a nucleus to contain their DNA. Depending on what type of eukaryotic cell it is, it could have different types of membrane-bound organelles. For example, a plant cell is likely to have chloroplasts, while an animal cell would not. Wow, look at all this alphabetized vocabulary. If you want to try to practice your skills, pause the video and see how many of these vocabulary words you can use to compare and contrast prokaryotic cells with eukaryotic cells. It's important to grasp that all cells of living things fall in one of these two categories. And understanding the characteristics of these two cell types can help us better understand the diversity of living things, whether they are archaea, bacteria, protists, fungi, plants, or animals. And in the case of my example, realizing whether an infection you're dealing with involves prokaryotic cells, such as bacteria, or eukaryotic cells, such as the fungus. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Thanks for stopping by. This is 2-Minute Classroom, and today we're comparing prokaryotic cells and eukaryotic cells. First, we'll talk about 5 major differences between them, and then at the end I'll give you some tips on how you can remember which is which so you can do well on your tests and quizzes. And if you really want to dominate your classes, be sure to subscribe to 2-Minute Classroom to bookmark my channel and see all my latest content. Now let's talk about the differences. Eukaryotic cells are made up mostly of organisms you are familiar with, like animals, plants, fungi, and protists. Prokaryotes are composed of bacteria and archaea. Prokaryotic cells are generally small, 1 to 10 micrometers, while eukaryotic cells are larger, 10 to 100 micrometers. And if you're wondering how small a micrometer is, 1 centimeter equals 10,000 micrometers, so they're really small. Prokaryotes are unicellular, meaning the entire organism is composed of a single cell, whereas eukaryotes can be multicellular, like plants and animals, or unicellular, like phytoplankton and protozoa. The biggest differences come inside the cells. Eukaryotes have membrane-bound organelles. These include the mitochondria, chloroplast, and Golgi apparatus. These structures have their own membranes and perform specific functions within the cell. Prokaryotes have no membrane-bound organelles, so no mitochondria, no chloroplast, no endoplasmic reticulum, etc. The final difference I'll highlight is in their genetic information. Eukaryotes have large amounts of DNA that is linear and stored in the nucleus. Prokaryotes have no nucleus, small amounts of circular DNA that's freely floating in the cell. So now you know the differences, but how are you going to remember them? Just remember that you are eukaryotic. You have eukaryotic cells. You are larger, you are multicellular, you have a nucleus, and you have a mitochondria. So when you see eukaryotic, just think of you, and everything else is prokaryotic. I hope that helps. Put any questions you have in the comments, and if you want to get more involved with 2-Minute Classroom, check out the community tab on my channel page. I love to connect with my subscribers and viewers, so please check that out. Here are some other videos about cells that you'll likely find helpful. Thanks for watching, and I'll catch you next time.
There are a lot of amazing theories in science. The general theory of relativity, atomic theory, the cell theory. And one thing we want to point out about a scientific theory is that it's a lot different from how you might use the word theory in your daily life. A scientific theory is not an educated guess. A scientific theory is an explanation of a scientific event, supported by scientific evidence. It must be testable and tested over and over and over again. And while theories can be changed or even disproven, you do want to realize there are a lot of facts behind them. One of our favorite theories of all time is the endosymbiotic theory. We love it because we just find the events in this theory to be amazing. The theory gives an explanation for how eukaryote cells could have evolved from prokaryote cells that lived in symbiosis. Prokaryote symbiosis means these organisms live together. So just to recap from our Intro to Cells video, when you think of prokaryote, think of pro rhyming with no nucleus, no membrane-bound organelles. Eukaryotes, on the other hand, remember eu rhymes with do, and they do have a nucleus, and other membrane-bound organelles. Both prokaryotes and eukaryotes are cells, though, and therefore they do have a few things in common that cells have, including a cell membrane, cytoplasm, ribosomes, and genetic material. But just look at these two cell types and you will see that one is way more simple. So what explanation does the endosymbiotic theory provide for how eukaryote cells could have evolved from prokaryote cells? A long, long time ago, seriously a long time, over 2 billion years ago, there were prokaryotes, much like there are today. They've been around for a long time. Well, just as there are in many organisms, there was variety in prokaryotes. Some of the prokaryotes had photosynthetic abilities, which means they could make their own food using sunlight energy. And those were the photosynthetic bacteria. Some of these prokaryotes were bacteria that had the ability to use oxygen to produce ATP energy. And some prokaryotes were larger, they could consume others, engulfing them. So obviously they had to be larger. The endosymbiotic theory is that some of these large prokaryote cells engulfed some of these small bacteria. But instead of those small bacteria getting digested, some of them remained intact within the large prokaryote, and they actually began to live as symbionts. That means they lived together. And for some of these cases, the larger prokaryote had engulfed the small bacteria that used oxygen to produce energy. This is believed to be the ancestor heterotroph eukaryote cell. And eventually, some of these cells engulfed the small bacteria that could do photosynthesis in addition. And there were some more living as symbionts. This is believed to be the ancestor autotroph eukaryote. Now, it's time to do a little reveal. We consider the bacteria that used oxygen to produce their own energy to have evolved into what is now mitochondria. And the bacteria that use sunlight energy to produce their own food, well, we consider those to have evolved into what are now chloroplasts. It's likely that these ancient eukaryotes had some kind of advantage in their endosymbiosis, and it's also likely that the mitochondria developed first, before the chloroplasts, because if you remember from our Intro to Cells video, nearly all eukaryote cells have mitochondria. It's just that eukaryote cells that can do photosynthesis can have both mitochondria and chloroplast. Now, if you remember our beginning about theories, you may be wondering, where are the facts for this? We'll give you a few of the facts. First, mitochondria and chloroplasts have their own DNA. Yes, separate DNA from what is found in the nucleus. Not to mention that their DNA is arranged in a similar way to prokaryote DNA, specifically bacterial DNA. The size of mitochondria and chloroplasts tend to be similar to the size of bacteria. And when mitochondria and chloroplasts divide, which they can divide all on their own independently, they divide in a way that is similar to how bacteria divide. So the endosymbiotic theory provides an explanation for how modern eukaryotes evolved from prokaryotes. There's obviously more questions you might want to explore. For example, what about some of the other structures and organelles in eukaryotes? Well, keep exploring. Secondary endosymbiosis is a great place to start. One last thing we want to make sure to emphasize. Endosymbiosis isn't just reserved for a theory that explains a past event in ancient history. No, because endosymbiosis is actually happening today with many other kinds of organisms. One of our favorite examples, the termite. Yes, termites can have prokaryotes that live in their gut and help them digest wood. And without them, well, let's just say digesting wood wouldn't happen so well. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.